Now here, along with the companion cells and the seed tube elements, you will have parenchyma cells. Those are called phloem parenchyma. Okay, let's show how the phloem parenchyma is actually helping the phloem in translocation of food. See here, parenchyma is acting as another supporting tissue along with the companion cells to the seed tubes. This is a longitudinal section. If you take the transfer section also, you are having parenchyma cells along with companion and tube cells. So all the seed tubes, companion cells, parenchyma cells, all these are living cells. Okay. So probably they won't have lignin. This is the basic difference between xylem and phloem. So what we talked in xylem, we are having major dead cells. Yes, except parenchyma we talked. Except parenchyma, all are dead cells in xylem. Whereas in phloem, almost all are living cells. Okay, except
growing fibers.
So in this way, a question can be framed also. They may give a list of, living cells and dead cells. And they may ask you to find out the odd one. So among the living cells, one dead cell may be given, or among the dead cells, one living cell may be given. Okay. So when you talk about phloem, the companion cells, the phloem parenchyma, the seed tube elements, all are living cells. That's why they doesn't have lignin. Okay. Probably they will have cellulose in their cell wall. But fibers, they are dead. Why fibers only are dead, that we'll see in the next slide. Okay. Now when you take the parenchyma cell, there's some point to understand about parenchyma cell. Now if you take a phloem, basically phloem or xylem can be classified as primary phloem and secondary phloem. We discussed about what does mean by primary and secondary in meristematic tissue and earlier classes. Do you remember the difference between primary nature and secondary nature? Primary, I told the initial stages of plant growth will find some structures. And in later stages of plant growth, we'll find some structures. Yes. So during the initial stages of plant, we'll find primary structures. As the plant growth advances, we'll find secondary structures. Just basic example, we can say stem, you take stem of trees. During the initial stages of plant growth, the stem comes in it. But in later stages, I mean in the secondary stage or in later growth, it becomes thick and becomes trunk. Yes, it's a basic difference. I showed the annual rings, I explained another class. Okay, so here primary and secondary terminology is related to the plant growth. So during the early development stage, initial development stage we call, what are structures we find, they called as primary structures, and with advancement of plant growth in later stages of plant life, what are structures we find, they are called secondary. Okay. Now here, why we are discussing about here with phloem, especially with the parenchyma, phloem parenchyma. There's a difference between phloem parenchyma at primary stages of plant and secondary stages of plant. When you take the phloem parenchyma, we will see axial type of phloem parenchyma and ray type of phloem parenchyma. Means what? Axial, longitudinal. You're familiar with this word in maths and all, isn't it? So if you have parenchyma cells arranged longitudinally, like this, side by side. They are called axial parenchyma. And if they are radially arranged, like this, the cells of parenchyma, then such parenchyma, that phloem we call as ray parenchyma. Okay, get the words ray and axial. How we are saying? Based on the arrangement of parenchyma cells. How the cells are arranged. The cells are arranged longitudinally, it's called axial. If the cells are arranged in the radial format, okay, that is called ray parenchyma. So here when you take the initial stages of plant growth, you will find only axial parenchyma. Yes? When you take the secondary growth or advanced growth of the plant in later stages, you will find ray parenchyma. That's the difference. So in initial stages of a plant, if you take, the parenchyma cells will be longitudinally arranged. As the plant growth advances, the cells will get radial arranged. This is one longitudinal line, another longitudinal, and the longitudinal. It makes a radial format. Okay. So this is the basic difference of phloem parenchyma in case of primary and secondary stages of plant life. Are you clear with these terms? Axial parenchyma, ray parenchyma, phloem parenchyma at the primary stage and the secondary stage. Any doubt with this point? Yes, let me know if you have any doubt regarding this point. No questions? People, are you there?
Yes, ma'am.
Do you have any questions regarding this? If you don't have any questions, I'll ask a question regarding this.
Ma'am, can you explain difference between ray parenchyma and axial parenchyma once again?
Fine. When you take okay, when you take the phloem parenchyma, we have two types of phloem parenchyma. We have axial parenchyma, then ray parenchyma. Okay, so this differentiation we are talking based on the arrangement of parenchyma cells. Suppose the parenchyma cells are longitudinally arranged. Means what? They are present side by side. Like this. If you have parenchyma cells arranged side by side, all the parenchyma cells, then this parenchyma we call as axial. Okay. Now suppose the parenchyma cells, they are arranged radially.
Yes, ma'am.
Assume like this. You're having arrangement of cells somewhat like this in the form of rays. Okay. So this we call as ray parenchyma. See, this axial parenchyma and ray parenchyma based on arrangement of parenchyma cells. That's all. See, this we can say as the axial parenchyma, isn't it? Longitudinally arranged parenchyma cells. All the cells are arranged longitudinally. Longitudinal companion cells. All the parenchyma cells. So this we can call as longitudinal axial parenchyma. Okay. Other than that, if they are arranged in the format of rays, it will be called ray parenchyma. And axial parenchyma will find only in primary stages of plant. And the ray parenchyma we'll see at the secondary stages or advanced stages of the plant. Okay,
Okay, ma'am.
Now my question is, why axial parenchyma is present in primary stages and ray parenchyma you will find in secondary stages of plant? Why can't the axial can be seen in secondary stages of plant? Got my question? So here we are saying this will be in primary stage and this will be in secondary stage, isn't it? So why? If you got the difference between axial and ray term, you'll get the answer. What we are saying is axial, the cells are arranged longitudinally. Means what? It is the simple one. You'll have transport in one direction or in two way, like this only. Translocation of food only, no? The function of phloem, parenchyma and phloem. When you take ray arrangement, in all directions you'll have advanced translocation of food material. As the plant grows, one direction of translocation may not be sufficient, because here I told what does mean by the primary and secondary small difference. When the stem at initial stage primary level it will be thin. Okay, so one direction of translocation of food is enough, sufficient. But when the stem gets thickened and it becomes trunk, if you have advanced translocation of food, then the plant will be healthy, isn't it? That's why you will have the ray parenchyma in secondary tissues and axial in primary tissue. This is an analytical approach regarding parenchyma, especially phloem parenchyma. Okay?